अस्सलाम वालेकुम स्टूडेंट्स। हम सॉइल केमिस्ट्री के लेक्चर को कंटिन्यू करेंगे और आज का जो हमारा टॉपिक होगा इट विल बी अगेन द ऑर्गेनिक मैटर कंपोजिशन एंड फ्रैक्शनेशन। ऑर्गेनिक मैटर क्या होता है ये हम ऑलरेडी स्टडी कर चुके हैं। कंपोजिशन इसकी क्या होती है, कुछ पार्ट हमने पहले स्टडी किया है, जो बाकी का पार्ट है वो भी हम यहाँ पर स्टडी करेंगे और उसके साथ कि हम इसकी फ्रैक्शनेशन कैसे कर सकते हैं। फ्रैक्शनेशन मीन्स how you can separate different components of the organic matter into different parts. Suppose I say in the soil or organic matter has 5 parts or 4 parts. How you will separate those 4 parts separately? How you will separate them? This is actually the fractionation. It happens due to chemical reactions. You proceed them within the lab, and let's start it how we will do. So let me figure out what will be the outlines here. First of all, I will explain you the equation for the loss and gain in soil organic matter. In the previous lecture, you remember we discussed about the addition of organic matter into the soil to build up the organic matter in the soil. So how you have to calculate, how you will estimate how much organic matter you have to add within the soil. We will try to understand this scenario. Another one is the organic components in the soils. What are the organic components? They are residing within the soil. And finally we will see the fractionation scheme for humic substances. So let's start organic matter balance. What is actually the organic matter balance within the soil? Here is a general equation for the loss and gain in soil organic matter. So change in the organic matter, here you can see change in the organic matter content of the soil is equals to the amount of organic matter added and minus amount of organic matter decomposed. You understand, I said you can add the compost in the soil, you can add biochar, green manure and the number of other organic amendments within the soil. So how much you are adding minus how much it is going to be decomposed in a limited time, in a specific time, and suppose we say the time is 1 year. So how much organic matter you are adding within the soil, how much it is going to be depleted or decomposed in the soil for 1 year. Ultimately you will just do a calculations, a simple mathematics, and you will find the change in the organic matter. Either it is built up, either organic matter is increased, or it reduced, it decreased, it depleted. What happened actually within that soil, particularly soil. So we will try to understand this. Let's find a case study. We suppose we have a time frame from the January to December, time period of the 1 year. What we will try to do here, we will add 40,000 kilograms of the humus, and we assume that annual decomposition rate is 2%. हमें ये मालूम है कि हमने 40,000 किलो, ओके, 40,000 किलो ह्यूमस आप सॉइल में ऐड कर रहे हो, और आपको पता है कि वहाँ से 2% के हिसाब से 2% के रेट से डीकम्पोजिशन हो रही है, डिप्लीट हो रहा है। जब 2% के हिसाब से डिप्लीशन होगी तो पीछे क्या रह जाएगा आपके पास? 39,200 किलोग्राम ऑफ द ऑर्गेनिक सॉइल ह्यूमस रिमेनिंग। ये आपके पास 1 साल में होगा, बाकी जो 800 किलोग्राम है उसने 2 साल में ख़त्म हो जाना है। अब एज्यूम करें कि 1 साल में आपने 2400 किलोग्राम क्रॉप रेजिड्यू ऐड कर दिए। ऊपर हम एज्यूम ये कर रहे हैं कि एक सॉइल में पहले से 40,000 किलो ह्यूमस मौजूद था, 1 साल में वो 2% डिप्लीट होता है, तो अल्टीमेटली आपके पास आ गया 39,200 किलोग्राम। जबकि अब आपने इस लेवल को अचीव करना है, एटलीस्ट आप सॉइल से कुछ ले रहे हैं तो उसको रिटर्न करना है। रिटर्न करना इज़ बेसिकली about the improving of soil quality, improvement in soil health. So you will add, suppose 2400 kilogram of the crop residues. Try to understand, these are the crop residues. These are not organic matter. It is actually the crop residues, the fresh crop residues. They are not the organic matter. They will get decomposed, they will be converted into organic matter, their mass will reduce. We have the deficiency of organic matter for the 800 kilogram while we are adding 2400 kilogram. You got it? That is about 4 times higher than the organic matter. So we assume that with the passage of time, 2 by third of the residues are decomposed and only one part is humified. So one part becomes 800 kilograms of the humus. So, so this 8 kilogram plus 39,200 kilogram, it will become finally the 40,000 kilogram. So if you are adding 2400 kilogram of the crop residues in the soil, actually you are not depleting the organic matter, you are not actually taking such organic matter, you are not actually damaging your soil, you are on stability. If you want to increase the organic matter, you have to change this value 2400. It must be higher, it must be higher, it must be like 4200 kilograms or something, anything greater than 2400 kilograms. I hope you understand this. Next, how you can say that the schematic representation of organic components of the soil. Let's explain here what are basically the organic matter components. Organic components of the soil, they always have two parts. One of them is the living matter. We call it the edephons, and the next one is the non-living, and we say it the organic matter. That's of our interest, key interest. Non-living organic matter also has two components, like unaltered material which is not changed, that is the stable one, and altered or transformed products, that is humus. Humus further has two components, non-humic substances. Non-humic substances, they will be amino acids, carbohydrates, lipids, lignin, nucleic acids, etc. While the humic substances, they have three components: fulvic acid, soluble at all pH values, light yellow to yellow brown color; humic acids, usually it has neutral pH; and alkaline pH, it precipitates at pH less than 2, dark brown to black, while humin is insoluble at all pH values, black in color. So these are basically some components of the organic components of the soil. Now if we fractionate the organic matter components, such components we just studied in the previous slide here in the last three sections, how we will do this? We will have a simple protocol that you will take the soil sample, you will add a strong base means which has the higher pH. It can be the sodium hydroxide with high normality, and you will shake it under the nitrogen, in presence of the nitrogen. Then you will have a soluble phase, and that soluble phase should be filtered. After that you will add the you will reduce the pH to 2 with any mineral acid. Obviously it should be the strong acid. The soluble phase remaining after acidification will be called as the fulvic acid, while insoluble phase will be humic acid. So here we have a very clear questions, we can discuss it. In the organic matter, when we fractionate, we always have two phases. One is the soluble, one is the non-soluble. So if there is a soluble phase, we call it fulvic acid, while insoluble part is the humic acid. These are operationally defined fractions of the soil organic matter. What is meant by the operationally defined fractions? Because they both of them, the fulvic acid and humic acids, they have to operate within the soil. They have their separate functions. They have to perform their specific duties. So that's why we are saying always them the operationally defined fractions. As might be expected, fulvic and humic acids have different properties to complement their differences in solubility. Now, you understand a very simple formula and thumb rule that like dissolves like, unlike dissolves unlike. So if there is a very soluble part, that is fulvic acid, it will dissolve a specific type of the compounds in the soil which are soluble in the fulvic acid. While humic acid, it will precipitate with sort of some other chemicals and it will not be solubilized. Fulvic and humic acids are mixture of different compounds of varying molecular weight and properties. One of them is soluble, other one is non-soluble. Both have different chemical properties, both have different masses, both have different affinities. So that's a simple chemistry actually. Fulvic acids are lower in molecular weight and more highly oxidized than humic acids. So lighter in weight, easy to soluble in water, easy to soluble in other parts of the soils, while humic acid non-soluble, highly oxidized. Structure of the fulvic and humic acids are unknown mostly to the researchers because they are complex. So if something is complex, if researchers don't know exactly right now, that's mean there is an opportunity. There is an opportunity, there is a question that should be solved, that can be solved. A good deal of the research has found that both polymers contain several important functional groups that influence soil properties and reactions such as cation exchange, CEC, you mean to say, buffering, chelation, and adsorption. So, as both of them they are within the soil solution, within the soil, they have some specific responsibilities, roles within the soil. For example, if the fulvic acid is there, it will perform such phenomena, such reactions which are related to the soil solution because it will be dissolved within the soil solution. While the humic acid, it can react with the soil minerals, it can react with the microbial part of the soil, it will affect the cation exchange capacity, it can provide the surface for the adsorption of the nutrients, adsorption of the organic compounds or something other. So both of them they have actually very good importance, very high importance, and they are very influential within the soil. So if I simplify everything here, that what is the classical fractionation scheme for the soil humic substances, what you will do, you will simply take a bulk soil sample, you will physically separate organic part. What is organic part means some roots, animal parts, feces, anything which you can separate with the sieve. Remaining soil organic matter will directly face the strong base, as I said, 0.1 molar. It will have two phases, soluble and insoluble. Insoluble will go to the human while soluble will go to the fulvic and humic acid mixture. Still it is mixture. It is, we usually have three parts, fulvic acid, humic acid and human. On the first phase you will separate the human, that will be insoluble, while the soluble will be fulvic and humic acid. Again you will treat with the strong hydrochloric acid and you have to maintain the pH 2. At that stages fulvic and humic acid will be separated on low pH. At higher pH fulvic humic acid on one side and humin on the other side. But if you want to separate the fulvic and humic acid, you have to apply the pH 2. So you, to apply the pH 2, you have to dissolve in the hydrochloric acid. So there will be two parts: soluble, insoluble. Insoluble will be the humic acid. You can further extract hematomilanic acid from the humic acid, and the soluble part will be the fulvic acid. So let's see, another thing is the important functional groups in the humic substances. As we said that we have the humics. Humic are actually the solid non-soluble part. So they have also some different compounds, organic compounds. They are very interesting. They have amino group: amide, amine, aldehyde, ether, ester, and alcoholic groups. Aminos very important for the amino, amides, amine, and aldehyde, ester. They are very important for the proteins, ketones. Another one is the carbonyl, carboxyl, enol, ketone, ketoacid, amino, amine, phenolics, imidazole, quinine, hydroquinone, and catechol. So all of these, they are very important for various chemical reactions happening within the soil, nutritional point of view for the plants, activity of the microbes within the soil, and their affinity with the plant roots. So all these are important. Thank you very much, guys. If there is any question, just go to the portal. You can paste the question there. I will respond, or you can just go to the YouTube channel. These videos will be there, and you can comment. We will communicate also there. Thank you very much. Stay safe.